my boots on and lace them up. A hard work, not another day to work. A hard work, that's what they say.
You get a sense of what words spring to mind when you hear evolution. Darwin? Okay, well, I hope that one comes after Darwin. What's that? Okay. Yeah? Natural selection. Change. So, yeah. So, nobody said the one I was going to say next. Dinosaurs. Right? I think most people on the street, if you ask them about evolution, they think dinosaurs, or they think, humans evolving from apes or something like that, right? But if you're not thinking dinosaurs or humans evolving from apes, you probably are thinking about, so Darwin is obviously the next person we think of. And that, in fact, was the first case. And we think of Darwin because I think most people, they think of Darwin, and Darwin seems like the first evolutionist, the father of evolutionary thinking. And of course, he did write the very influential On the Origin of the Species. But it's also important to remember historically that he wasn't the first person to think about evolution. And after all, there was Lamarck early in the 19th century. His grandfather, Erasmus Darwin, actually also was thinking about evolution. So if you think about it, what his real contribution wasn't the idea that evolution happens. It's really, most fundamentally, the idea that all living things are descended from a single common ancestor. So Lamarck's evolutionary thinking involved change over time, which is fundamentally what evolution is. But he didn't have the idea that all things, plants, animals, fungi, bacteria, whatever, evolved from a single common ancestor. And so that's probably Darwin's most fundamental insight, and the most important thing that he contributed with respect to evolution is that single idea. Beyond that, there are a couple of other things that I sort of think of as his grand ideas. And I keep saying his grand ideas with respect to evolution because any of you who know anything about Darwin realize that he made, even if he had never written On the Origin of Species, we'd still revere him very highly as a biologist. He figured out phototropism in plants, the fact that plants grow towards light and towards gravity. He and his son did experiments on that. He did all sorts of experiments and observations on pollination in plants, on the expression of emotions in animals, and so on. And so he did a tremendous amount of things besides evolution. But within evolution, he had two grand ideas. We heard one of them already, namely natural selection, the idea that one of, and I say one of, not the only, but one of the important mechanisms by which evolutionary change happens is the process of natural selection. And the second grand idea is related to his fundamental insight that all living things are descended from a single common ancestor. That's the idea of descent with modification. So that new species arise, they are similar to, but not identical to, the species from which they evolved. And it's that gradual accumulation of differences over time that's responsible ultimately for the entire diversity of life that we see, plants, animals, bacteria, and so on. And so I have to throw in a couple cartoons, and this is actually sort of an educational cartoon taken from this fabulous website, if you don't know, at UC Berkeley on evolution. There's a fabulous resource for teaching and study for students who are learning evolution, illustrating the process of natural selection. You start with a mixture of the green beetles don't show up as too green here, but a mixture of green and brown beetles. The birds eat the green beetles. Since the green beetles have green offspring, there aren't too many green offspring in the next generation. There are more brown offspring. Over the years, then you get natural selection, so you have evolution of brown beetles. That's the fundamental idea of natural selection. Now, both descent with modification and natural selection require something that we often forget and don't teach as well as we should when we're teaching this to students, and that is it requires heredity. It's not enough for the green beetles to be eaten and the brown beetles not to be eaten. unless it's also the case that brown beetles tend to have brown offspring and green beetles tend to have green offspring. Otherwise, all you've done is you've sorted things in one generation, and then the next generation will just be the same thing starting all over again, right? So you need to have that resemblance from one generation to the next. That, there is also at least one really big mistake Darwin made, one grand failure. And although he recognized, as any intelligent person would, that offspring resemble their parents. I mean, after all, one of the things he did as a hobby was breed pigeons. And it had all these different breeds of pigeons. And you sort of knew what the genealogy of pigeon breeds was, tumblers versus rollers versus all that stuff. So he knew that offspring resembled parents, and he understood that resemblance was vital for both his mechanism of evolution by natural selection and descent with modification. But he proposed a mechanism that was completely wrong. Just out of curiosity, how many people have heard of the word pangenesis before? A few. Yeah, so he had this idea that all parts of the body of a plant or an animal somehow had these little things inside them called gemmules, and those gemmules would all fuse somehow into the sperm and the egg that were transmitted to the next generation. And so we often associate Lamarck with the inheritance of acquired characters. You know, the giraffe strives to have a long neck, and so then its offspring have. Well, in fact, Darwin also thought that there was inheritance of acquired characters, and gemmules were the mechanism he thought that was produced that. And so I mention this just to remind us all that, yes, Darwin was a brilliant scientist, had fundamentally important and earth-shattering insights. He also got things wrong because he, after all, was human. So I just wanted to throw that in. So again, fundamentally, his most important insight, arguably, is that all living things are descended from a common ancestor. And to put this in some perspective, I just pulled this off the web to illustrate what we mean by all living things. So that's a tree of life. That's interesting, the perspective is different on this screen. So it's a nice circle on my computer screen. It's sort of stretched out here. But anyway, you can see what's going on. And so that does represent all diversity of life. You can see it's sort of color-coded by major groups. Now, here's a test. How of that whole circle, and Janine, you can't vote, because you'll know the answer. Of that whole circle, how much of that circle do you think is accounted for by plants? But, you know, sort of pine trees, ferns, flowering plants, things like that. Very small. Okay, it's definitely the little stuff. But it's not even the little stuff that you study. Yeah. So see that little gray bar right there? That is all green plants. Okay? How about animals? Well, I don't know. We have a zoologist here. Yeah. Oh, yeah. Insects. Multicellular animals, and including insects, sponges, tapeworms. You'll hear about tapeworms later. That's why I'm giving Janine such a hard time. As well as vertebrates. So it is bigger, but it's still pretty small. Well, actually, I think that represents this is, of course, constructed from DNA sequences sometimes, and that represents sort of the sequences that are available. If you could read the names there, you would see. Let me use my cursor to point to it. You would see there are two names right here adjacent to one another that are both Caenorhabditis elegans and Caenorhabditis something else. So they're two little worms that are very similar to one another. They're used a lot in developmental studies. We have both Pantroglodytes, chimpanzees, and humans, which are basically the same thing, except one has a lot more hair. So I think the size of those doesn't necessarily represent the numbers of species, but what it does represent is if you look where plants and animals, which is what most of us think about all of the time, lie in the tree of life, relative to the depth of the tree of life, it's a tiny fraction of life's diversity, even though it's a really interesting and important diversity to all of us, because it's a part of diversity we are. We're just that. So we sit, just for your reference, right there. That's us. Oops, wait a minute, that's chimps. We're right next to that. Anyway, so that's us. And so that seeing that kind of diversity and that kind of depth of diversity, I think one of the fundamental questions that most evolutionary biologists are interested in one way or another, even though none of us can really grasp the whole big question, is why are there so many different kinds of species in the world? And so what I want to do for the rest of this morning, and actually I think what you're going to discover is I think Carolyn allotted me time till 10 after 10 or something like that. I don't anticipate actually taking that long, but what I want to do is to share with you for the next 20 to 30 minutes some of the insights I've had in the work I've been doing most recently for the last 8 to 10 years in South Africa on a really fascinating group of plants that gets at some of these questions and illustrates sort of the types of questions that evolutionary biologists try to study. But before I get there, I just want to back up, and we did hear about Galapagos Islands. Of course, the reason we hear about the Galapagos is also because of Darwin's finches. Now, you might know that although Darwin collected specimens of all these finches, which actually turned out not to be finches, but that's another story. Although he collected specimens of these during the voyage of the Beagle and took them back to Great Britain, he didn't really understand that they were different species or what any of the significance of those collections until he had taken them back and an expert ornithologist at the British Museum was looking at them and began to realize that in fact he collected different species of birds. And then he started to identify them, and Darwin would compare the species identifications that the expert had made with the islands from which he'd collected them, and he realized that they were in different places. And so that was one of the fundamental observations that led him to the idea that, in fact, there is descent with modification. The obvious way to explain that diversity is that there was a single ancestral bird that at some point in the past made it to the Galapagos Islands, and when it found itself there, diversified into a variety of habitats that were available because other birds weren't there occupying those habitats. And that's sort of, on a very small scale, the insight that leads ultimately to the recognition that all living things are descended from a single common ancestor. Now, I bring this up because it's sort of a canonical example of one of the evolutionary phenomena that I'm going to talk about, which is evolutionary radiations. So we have this warbler finch, in this case, which is a Peruvian species that is the closest living ancestor of the Galapagos finches. All of those are descended from a single common ancestor over there. In a relatively short amount of evolutionary time, they've diversified and now occupy a wide range of habitats and function in a wide range of evolutionary roles. And we actually understand quite a bit about what's underlying this diversification, both in terms of the fact that associated with the different beak morphologies, there are different feeding strategies, and with respect to understanding some of the actual molecular and physiological mechanisms that produce those differences. So in particular, it turns out that these differences in beak morphology, in particular between this large ground finch that feeds on very heavy seeds, large seeds, and this cactus finch that probes cactus fruits. You can see we have a very large, heavy, short beak here, a relatively light, slender peak there. It turns out there are just basically two different growth hormones involved in determining the length of the beak. That's calmodulin. And another one, this BMP4, that's involved in determining the width and the depth and the height of the beak. And so by looking at the relative expression of just those two genes, the authors were able to explain this variety of beak widths and lengths across Galapagos finches. And so we now have a really nice story in which we understand at a fundamental genetic level what genetic changes lead to the differences between these species, as well as understanding the ecological role that those differences allow the finches to play. And so that's the sort of thing that evolutionary biologists are looking for. In plants, one of the most famous examples of this kind of evolutionary radiation is in a group referred to as the Hawaiian silver swords, because of this iconic species here. If I've never actually made it to Maui, although I've been to Hawaii once, and so I was on the Big Island and on Kauai, but not on Maui. But this is if you go to Maui and Haleakala Crater, one of the plants you'll see there is this silver sword, which are just magnificent plants to look at. It and that sort of scrubby looking thing are in fact part of the same evolutionary lineage that arose relatively recently, namely about 5 to 6 million years ago, and diversified widely throughout the Hawaiian Islands in relatively dry habitats like this, as well as bogs and rainforests. Now, so on the one hand, this has been a tremendous evolutionary radiation with plants occupying a wide range of different habitats that are suitable to them, and so there's some adaptation involved. But what you won't be able to see, probably even when I point it out to you, is there's a scrubby little green bush back here. That scrubby little green bush is this guy. So in spite of the fact they looked very different, they're actually able to survive and do pretty well in the same environment. So there's something else going on there besides just adaptation, or perhaps at least they are adapting to the same environment in very different ways. The other thing that's even worse is this evolutionary radiation has happened so recently, those two things can actually hybridize with one another and produce really weird looking offspring. So another example of an evolutionary radiation that's been nicely studied is illustrated here in a group of plants. Some of you may have these growing in your garden, columbines. And so you can see here, the focus here is on how the plants have differentiated with one another with respect to the pollinators that transfer pollen. So the light blue things with a relatively short nectar spur at the top are pollinated primarily by bees. The bright red things are pollinated primarily by hummingbirds. And the ones with very long nectar spurs with sort of pale blue and white corolla are pollinated by hawk moths. And the blue, red, and yellow on this phylogeny illustrates the typical pollinator syndrome associated with different species in this genus. And again, all of that diversity has arisen relatively rapidly. In this case, again, they're working out the genetic basis of the differences in nectar spur length and so on, and relatively simple genetic changes that increase the nectar spur length lead to dramatic diversification with respect to pollinators. So what all of those examples illustrate then is that evolutionary biology can be divided, in the way I think about it at least, into two broad categories of study. We're either studying the pattern that evolution has produced or the processes that have produced the pattern. And so by pattern, what I mean is the history of life on Earth. The branching pattern of how species are related to one another, the time at which they arose, the time at which they went extinct, and the sort of the chronological history of what happened on Earth, who's related to whom. By process, what I refer to is a study of the mechanisms that are responsible for producing that pattern. To a large extent, until the last 10 to 15 to 20 years, maybe, evolutionary biologists have tended to focus almost entirely on either one or the other of those two pieces of evolutionary biology. So you have, I mean, and so the sort of, in a way, the canonical example of people studying pattern might be those people who study the evolutionary relationships among dinosaurs or other extinct groups. So they go out, they dig up fossils, they figure out how those fossils fit into the tree of life, and they know sort of the sequence of origin and extinction of plants and animal groups and how those are related to one another. Or even contemporary biologists studying existing organisms may focus on pattern by trying to use characteristics of the morphology, anatomy, DNA sequence to reconstruct the pattern of relationships. And both of those can, and in the past, often did proceed without trying to say anything at all about what the underlying evolutionary mechanisms were. And not just in terms of what are the genes that produce the morphological differences, but were the processes ones in which natural selection is responsible for the difference, or were they processes that arose, where the differences arose simply as a result of species being isolated in different places and differentiating randomly with respect to one another? Because that kind of random differentiation can also play a very important role in evolution. And so that sort of leads to what I'm interested in. So for the last 10 years or so, I've been very interested in a particular region of the world known as the Cape Floristic region. Now, to set this in context and tell you why it's a really interesting place for a biologist, and especially for a botanist to spend a lot of time, I put up this list of biodiversity hot spots around the world that was identified actually almost 15 years ago now by Norman Myers and colleagues in Nature. And what they've identified here is a relatively small number of places in the world where most of the world's biological diversity is found. Their objective here, they weren't doing this for evolutionary purposes, they were doing this for conservation purposes. They were interested in identifying, if we are really concerned about preventing extinction and conserving as much biodiversity as possible, where ought we as concerned citizens to devote our effort? And so what they did is they identified those areas that had the highest concentration of species that were unique and found nowhere else, and then identified those as hotspots. And so you can see we've got only one in the US, the California Floristic Province, Mesoamerica, the Brazilian Cerrado, and so on. The one I want to draw your attention to is the one right down here in the southwestern part of South Africa, the Cape Floristic Province. That's where I've been working for as I say, about the last 10 years. Now, just as an aside, before I get into talking about the evolutionary biology here, I should say, since I suspect many of you are also very interested in conservation, preventing extinction, and so on, that in fact, although on the face of it, this hotspot approach seems like a very logical way to go, there's actually a fair amount of controversy about the extent to which we should focus on those hotspots. Because you'll notice, there's nothing in the North Temperate region. It's like saying we don't care about boreal forests at all. So, everybody who's been worried about drilling in the Arctic National Wildlife Refuge, well, it doesn't look like you need to worry, because it's not a hotspot. So we can talk about that later if you want. That's not what I want to focus on. Let me tell you why I think the Cape Floristic region is really interesting and important. First of all, notice, it's a little hard to see, but there's actually two hotspots here. There's the Cape Floristic Province, and then there's the succulent Karoo in South Africa. And just in terms of how big they are geographically, both of those are a lot smaller than any of the other places that are recognized as hotspots. So that already tells you something interesting. It tells you that in a pretty small geographical area, there is a large amount of biological diversity. Now, to make that more specific, 90,000 square kilometers roughly, which probably doesn't mean anything to per se other than that, well, that seems like sort of a big number, but you don't have any sense of scale. As a sense of scale, that's about the same size as the state of Maine. So we're talking about an area about the same size as the state of Maine that has 9,000 species of flowering plants. Now again, unless you're a botanist, the 9,000 may not sound terribly big. To put it in perspective though, keep in mind there are only about 7,000 birds in all of North America. 9,000 species of flowering plants is also more species of flowering plants than you'll find in floras that cover the northeastern United States as far south as the Mason-Dixon line and as far west as the Mississippi River. So we're talking about a very large number of species. In fact, the level of species diversity in the Cape Floristic Province is on the same order as the most diverse tropical rainforests anywhere in the world. So it's a very diverse region, and so therefore a very interesting place to ask that question about why is it that there are so many species in the world? Because this is one place where there's been a tremendous explosion of species diversity. The other thing that makes it interesting is that about half of those 9,000 species are found in a relatively small number of closely related groups, about 30 clades or monophyletic groups. Proteaceae, the family Proteaceae, happens to be one of those groups in which there are a large number of species. It's one of the 10 largest families in this region. Two genera in particular, the genus Leucodendron has about 82 species. The genus Protea, which is the one I will be focusing on, has about 69 species. The genus as a whole has somewhat more than that, about 90 species. But nearly the vast majority of species are in the Cape Floristic region, and the species elsewhere seem pretty clearly to have evolved from species in the Cape Floristic Province. So they sort of escaped from the Cape and moved elsewhere in Africa. So the genus Protea is also quite young. Now, you'll notice there's a huge amount of uncertainty here in terms of how recently arrived, like a factor of 3. And there's a couple reasons for that. First of all, you may know that a lot of these estimates about how old groups are now based on the extent to which DNA differences, how much difference there is in DNA sequences between things. Well, the problem is that kind of a molecular clock works only if you can calibrate it somehow, right? Because the amount of sequence difference that corresponds to a million years between species of bacteria or tapeworms or fruit flies may be quite different from what we see in flowering plants, especially these that are fairly long-lived. And so there are two reasons why that's really uncertain. One is that there was a relatively small amount of sequence information available when the estimate was made. But the other thing is there is only one fossil that can be used to date the entire phylogeny of the Proteaceae. And that fossil lies somewhere way down off here. And so although the date of that fossil is relatively well known, projecting from there to the base of Protea adds a great deal of additional uncertainty. But in any case, it's relatively recent, the last 5 to 20 million years or so. And all of that diversity has arisen since then. Now, I've highlighted this blue clade because that's actually a group of species that I'm going to focus on for most of the rest of the time here. It's a group known as the white proteas. It's a well-supported group, clearly monophyletic, based on the calibration at the root of the tree of being 5 to 18 million years ago. You can project up and say this group probably diverged from its closest relative somewhere in the last 350,000 to 1.2 million years ago. So again, this is even more recent. We're talking about pretty recent divergence. It also, one of the nice things about this clade is although it's relatively quite recent divergence, it actually covers much of the geographical range that Protea does. So we've got a map of Africa here. The box shows you what's blown up. Just to orient you a little bit, Cape Town sits right about here, if you happen to know where Cape Town is. Johannesburg is just up there, just sort of up a little bit off the top to the right. So that's sort of we're in southernmost South Africa. This right here defines the boundary of the Cape Floristic region. And so even though this is a very small clade within Protea, its species cover most of the available habitat in the Cape Floristic region that's suitable for Proteas, as well as extending beyond into the Eastern Cape and beyond. Which is, so even though this is one example of an escape from the Cape, the major escape involved a completely different clade that I didn't highlight. And so we were interested then, given that, to look at a variety of evolutionary patterns and processes within the group. In particular, if you look closely, you'll notice this color right here, this sort of dark green color, shows up here and it shows up here, with a lot of other stuff in the middle. That dark green color corresponds to what had been described as a single species, Protea mundii. And before I ever went to South Africa, I saw that distribution map, and I said, you look at that and I say, that just does not make sense to me. You've got this small group that's more or less continuously distributed. How could it be that you've got this species here and the same thing there? And there are two, maybe more than two, but at least two possible explanations that immediately occur to me. One is, well, you know, maybe it really is the same species, and it recently dispersed over here to the western part. And I say that because we have a lot more populations here than we do in the western part of the range. And so it'd be most likely that the dispersal will go that way. The other possibility is the taxonomists just got it wrong, that they look the same, but they are really, in fact, different. And so one of the things we've done is to try and examine that question and figure out, well, what's actually going on here? Is it the case that these are really the same species and it represents some kind of a dispersal event? Or is it the case that they are, in fact, evolutionarily independent? And so the way we investigated that was to use some molecular markers that are known as microsatellites. These are very useful markers that are quite widely used today, even as we're beginning to use much more sophisticated and expensive means to sequence entire genomes, because they're still relatively cheap and easy to do. What they consist of is just very short, repeated sequences of DNA, like AT, 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 and they mutate or change very rapidly, because you can imagine when the DNA is replicating from one generation to the next, it'd be very easy for the polymerase to slip just a little bit, and it's like banana instead of banana, right? You just sort of repeat the syllables, and so you could very easily get differences in the number of repeats, and that's actually what we score with microsatellites. Is the sequence doesn't change, what changes is the number of repeats we find at a particular place in the genome. And because it's so easy for that number of repeats to change, they change very rapidly, so they're very sensitive indicators of recent evolutionary change.
What constitutes very rapidly?
So the best estimates of mutation rate are that, say, 1 in 1,000 replications, or maybe even 1 in 100 replications, will lead to a repeat difference. Whereas if you're looking at changes in nucleotide sequence, it's on the order of 1 in a billion replications. So it's orders of magnitude more rapid. And typically, the changes also don't result in changes that would affect the ability of the plant or the animal to survive or reproduce. And so they don't affect fitness, and so the only processes affecting their persistence are random processes of genetic drift and so on. And so what we did then is to collect leaf material from 36 populations, about 20 individuals per population, and we'd identified 10 different regions in the genomes that had these microsatellites, so we could score their allelic state and then look at them. And after a lot of statistical hocus pocus, what we come up with is something that looks like this. Now, this is a pretty complicated diagram, so I'm going to spend a little time walking through it, although the take-home message in the end is really simple. So if we start over there on the left, the 3, 4, 5, 6, 7, 8, 9, if you look really carefully, you'll see that corresponds to the number of colors in the bars going off to them, right? So the top bar you have pink, orange, and blue. The next one you add a green. The next one you add a red, and so on, okay? And so what's important is not that the number of colors per se, but those colors represent different clusters of individuals that into which the program has classified them, okay? So the next thing to look at is each little bar here corresponds to a single individual in our sample. So remember, we had about 20 individuals per population, about 36 populations. So we have roughly 720 individuals. And so there are 720 individual bars here. And the color assigned to an individual indicates the genetic cluster to which that individual belongs. Okay, and so the important thing about this analysis that's really interesting is that the program assigns individuals to those genetic clusters based purely on the genotype that we've scored for them. It knows nothing about where we collected them. It knows nothing about whether they were collected the same place as that plant or not. All it knows is I've got this genotype and it belongs in this cluster. And so the black lines here indicate the populations from which we collected them. But we just added those black lines later. Those weren't actually part of the analysis. And so the first thing you see when you look at this...
So is that a geological population?
Yes, so the black corresponds to a geographical location where we went out and collected them. And it's sort of loosely defined because it's not like we picked 10-meter square plots to sample them, but the populations are actually physically isolated from one another, so they represent geographically distinct clusters. Yeah. But so one of the first things you notice is that to a large extent, the geographical clusters are consistent with the genetic clusters. I mean, the most striking one is this subvestita here, is just solid blue, and you don't see that anywhere else, right? So that's the first thing. Now, the 3, 4, 5, 6, 7, 8, and 9, that's just a question of how many clusters we asked the program to identify for us. And there are various criteria you can use to determine what's the best number, which we won't talk about. What I want to focus on is I've also got the species names up there on top. We can ignore the hybrid part. You'll notice this P. mundii is the one I pointed out that has an eastern distribution and a western distribution. And so if we focus on that, you'll notice the eastern populations are pretty much orange. The western populations are pretty much green. And so what that says is the microsatellites are telling us these are actually evolutionarily distinct populations. Now, morphologically, in terms, if you look at their flower traits, their leaf traits, they're very difficult to tell apart. Once we know that, in fact, the microsatellites are telling us they're evolutionarily distinct, we go back and look at it, we can detect some morphological differences. So, in principle, you could recognize a specimen as belonging to one group or the other without knowing where it was collected, but it would be pretty difficult unless you had the DNA. But the bottom line is then we got a pattern in which largely the species that had been previously recognized were distinct. We detected some hybridization, and I didn't really point that out, but if you looked really carefully, you might have seen some places that looked a little funky. Those were hybridization. Most importantly, for what I was talking about with respect to understanding the pattern, is what we've identified here is a case in which those two populations actually seem to represent distinct evolutionary units rather than a dispersal from one place to the other. So not only then have we discovered something about the pattern of relationships within this small group, we've excluded a potential explanation, namely dispersal as being responsible for part of the pattern. And instead it reflects, presumably, some geographical differentiation elsewhere. Okay, so that's sort of the pattern part, primarily. Another thing that makes this region really interesting that I haven't mentioned yet is that there's some very strong environmental gradients. And in particular, there's a very strong environmental gradient with respect to both the amount and the pattern and timing of rainfall through the season. If you happen to know a little bit of geography, you'll recognize that latitude as similar to the latitude in Western Australia or central Chile, or if you made it a positive number, California or the Mediterranean. And it's on the west coast of a continent. That is, in fact, characteristic of a Mediterranean climate region. So think about California. That's about the same as Santa Barbara. They get rain in the winter, and it's dry all summer. So in California, rain starts sort of typically mid to late October. You have rain from then through March, maybe early April, and it's dry all summer. Here, it's exactly the same thing if you're around Cape Town, except that the season is reversed, of course, because we're in the southern hemisphere. So the rains start in sort of late April, early May, go through September, maybe early October, and then it's dry from October through March. That's what happens over here. As you move east and get into the central part of the Cape Floristic region, the rainfall is spread more evenly throughout the year. There also tends to be somewhat less, and what occurs is spread more evenly. When you get into the eastern part of South Africa, where this species is, the rainfall is again concentrated, but it's actually concentrated in the summer rather than in the winter, because it's associated with summer thunderstorms. And all this has to do, of course, with the global weather patterns, how air circulation is moving around, so that we have thunderstorms in the east in the summer. So there you have rain when it's hot. Here you have rain when it's cool, and you don't have rain when it's hot. But proteas have evergreen leaves that persist through for several years, and so they have to be able to deal with summer droughts in the Western Cape. So one of the interesting things then is, again, this is just this small group. This is a photograph showing you leaves collected from garden-grown plants of the different species, and if you look carefully, you'll notice there's sort of different letter combinations for some of these, indicating different populations of the same species. And there's not a huge amount of variation here. They're all basically sort of elliptical leaves. But on the other hand, these are all the same age, and they go from some that are very small in Venusta to some that are quite large, some that are pretty narrow, to some that are pretty fat. What you can't see so well there is they also differ quite a bit in how tough and leathery they are. And so there's a lot of variation in leaf morphology here. And one of the things we were interested in then is trying to understand, well, we've got all this variation. What process produced it? Is it adaptive? Does this represent a process whereby natural selection is optimizing the leaf characteristics, the leaf traits in a particular location to the climatic and environmental conditions of that place? Or does this just represent random differentiation that happened because the populations are isolated from one another and no longer changing genes? And so what we wanted to know is this variation adaptive? And to do that, you actually would have to do even more than what I've got outlined here, but you have to do at least these three things. First of all, we'd need to know, well, is there any sort of an association between those leaf differences and the environment where the plants occur? Because if there isn't, well, then there's no evidence that there'd be any adaptation. There's just the environment varies but the way the environment varies is unrelated to how the leaves vary. There's just no adaptation of the leaf to the environment. It's just varying. But of course, that's not enough, because as I said several minutes ago when we were talking about natural selection, in order for natural selection to work, the variation on which it's working has to be heritable. You have to have evidence that the offspring actually resemble their parents. So we need to know whether that variation is inherited. And then those two things get you quite a bit of the way there. But to really nail it down, what you'd really like to know is, can we demonstrate that the differences we observe are related directly to differences in performance? Is it the case that plants that have heavy, thick leaves actually survive better than those that have thin, floppy leaves under certain circumstances, for example? And so that's what I'll run through here very quickly. The first one is pretty simple. Again, this is just that small group of proteas. several different environmental variables we looked at relating to how cold the winters are, how dry the environment is overall, how concentrated the rainfall is, and then how fertile the soil is. And you can see in 3 out of the 4 cases here, with respect to particular traits we measured, we found pretty strong relationships. Now, I should also, in the interest of complete honesty, I should also point out that those dark red lines I've drawn in there aren't the actual regression lines that we used in the analysis, because it was a multivariate regression, and so it's a lot more complicated than that. But that's just to illustrate we do have those sorts of trends. Now, what's interesting is in the last several years, I've been working as part of a much larger group doing similar sorts of analyses across the entire genus Protea in the CFR, and also the genus Pelargonium. And I don't have the results for Pelargonium to show you, but I do have the results to show you for traits, for a single trait and 6 different environmental variables for about 50 different species collected from a little over about 70 some different sites in the Cape Floristic region. Every one of those points represents an individual leaf that we measured someplace. So we measured a lot of leaves. And 6 different environmental characteristics. And again, you can see there's, to varying degrees, some sort of an evolution, a relationship between this leaf mass per area, which is a measure of how thick and tough the leaves are, and these environmental traits. This is consecutive drought days is a measure of how long the summer drought is. The longer the summer drought, the thicker and tougher leaves are. So yes, as a matter of fact, we do have evidence that variation is associated with the environment. Okay, so that's sort of the first check mark. The next question is, well, is that variation actually inherited? And I'm not going to show you data, I'm just going to show you pictures of how we go about doing that. And so what we did is, in that first study on the smaller group of proteas, at the same time we were measuring traits in the field, and I say we, what I really mean is Jane, my postdoc. Jane collected seeds from plants in the field at the same time, brought those seeds back to a lath house in Kirstenbosch Botanical Gardens in Cape Town. And here what she's doing is watering seeds that she's germinated. And so each one of these little cells here contains a seed of a protea that was collected in the wild in some population. And so the point here is that when we measure the traits of the plants in the field, they might differ from one place to another, either because the plants are genetically different, or one place just happens to get more rain, and when plants get more rain and more water and more fertilizer, they grow bigger, right? And so we want to control for that. So we're bringing them all back to a garden in Kirstenbosch, and then we plant them out and grow them there and measure the traits in the garden. And so the traits won't match what they were back in their native habitat, but at least the plants have all been grown in the same environment, so if we see differences there, we know those differences are genetic. And so the take-home message from this analysis is we find evidence that those traits we measured, the differences are genetic, and the differences we see in the garden are consistent with the differences we saw in the field. So the relationships between traits and environments do reflect actual genetic differentiation, as well as perhaps some just plastic response to the environment. So yes, we've got variation inherited as well. The last bit then is to try and figure out, is it in fact related to performance in some way? And we did a couple of different things here. I'm only going to talk about one of them. But again, we, meaning Jane, when we were in the field, she looked at, for the time being, let's just focus on the little cartoon here and there. When she was in the field, we know the characteristics of the plants that we measured in terms of, in this case, we're looking at leaf area as one of the traits we're interested in. We could also count the number of seed heads on the plant, because proteas actually retain their seed heads for a very long time. And so that gives us a very good idea of how many seeds a particular plant has produced. And so we can look, and in areas where there's the rainfall is relatively aseasonal, larger leaves are associated with more seed heads per plant. So that would suggest that in areas with aseasonal rainfall, large leaves is evolutionarily advantageous, right? On the other hand, in areas where we have highly seasonal rainfall, large leaves are associated with fewer seeds per plant, suggesting that in those areas, selection is going in the opposite way. Okay? Now, what we've got, so that explains sort of what the selection looks like within a local population. Now, we summarize that selection within a local population by looking at the slope of this line. Both, is it positive or negative, and then how steep it is. And that's reflected in what we've got. Each one of these points here reflects the slope of a line estimated in a particular population. So the positive slopes on the left means that it looks like this. The negative slopes over here mean it looks like that. This dotted regression line is a regression line to those slopes, indicating that there's actually variation in selection across the environment. So we can actually predict we can actually predict which traits are going to be favored in which environment based on that kind of a regression. So the bottom line, we've got large leaves favored here, small leaves favored there, and so in fact, we have variation that's related to performance. So that's one really interesting set of features that we're now getting a good understanding of the processes that are responsible for producing the diversity. There's another really interesting aspect of diversity that I wanted to mention before I finish, and that is proteas have really spectacularly, most of them have very large flower heads. The king protea, actually, the flower head's almost a foot across. It's the national flower of South Africa. The rugby team is named the Proteas. They're really crazy about, there's a Protea hotel chain. They're crazy about Proteas in South Africa. They're also grown horticulturally because they're very beautiful. Some of them, as you can sort of see here, it's a little washed out, but some of them have two color forms, a pink and a white. Others have only pink. And what's interesting here is I've boxed in there the pink and the white symbol indicates that's a species that's polymorphic, that has both forms. The solid red indicates they have only the red form. And what you'll notice there is we have some very closely related species, one of which is polymorphic, the other of which is not. And so one of the things we're starting to do now is to try and understand the evolutionary dynamics that are responsible for why is it that some species vary in a trait and others don't. It turns out it probably has absolutely nothing to do with pollinators. It turns out instead it probably has to do with seed predation and perhaps the association of the thing that produces color in the flowers with protection against UV radiation at high elevations. But that's a story for a different day. So the processes then we've talked about, and we talked some about geographical isolation, natural selection, and adaptation. Those produce the patterns that we're interested in. And what I want to say in just the last few words is a little bit about what's next, where we're going from here as part of a large project involving 6 principal investigators at 3 institutions, including one in Australia. And what's going next is a couple of things. First, with respect to the pattern, you can see that it wasn't evident in that phylogeny of proteas I showed you earlier, but there's actually a lot of uncertainty about the precise relationships. We're going to really nail that down in the next year by using some state-of-the-art sequencing technologies to produce between 300 and 400 gene sequences from each individual plant we sequence. We're going to sequence 60 species, about 3 individuals per species. So we're going to have a massive amount of data there to really nail down the phylogeny in high resolution, so we'll be able to narrow the bounds on the time at which these different events occurred, and then also to understand the evolutionary history of the traits that we've been studying. Another thing, also taking advantage of the modern high throughput technologies, is looking at genomics, at the processes of evolution. And there's sort of two pieces to this process examination. One, first of all, we're going to be looking at between 10, instead of just 10 markers per genome, as we did with microsatellites, we're going to be looking at somewhere between 10,000 and 50,000 markers per individual plant for about the same number of plants, roughly 700. 19 populations, 30 to 40 individuals per population. That kind of data then will allow us to look both at the genetic basis of the trait variation we're interested in, using the same kind of techniques that human population geneticists use to identify the association between genes and high blood pressure or genes and diabetes. We can look at the association between genes and leaf width. And also, we'll be able, because of the huge amount of data we'll have, to get a very good understanding of the evolutionary relationships, not just of a single species to other species, but of these populations to one another, to get a much better sense of how the evolutionary dynamics is working within species across the entire landscape. Thank you very much. Yeah.
With this multinational cooperation
Yes.
collaboration going on, how do you share that massive amount of data?
Well, so the massive amount of data mostly sits on a server in Canberra, and then he pre-processes a lot of it to a smaller amount that is relevant for the later analyses, and then we deal only with that smaller amount.
Are you using Dropbox? Are you using home drive?
So the amount, he's got an FTP server, and when we need that, we just log on and go away and come back the next morning, because it is, it's not terabytes, but it is several gigabytes that we need to download from Canberra.
Can you, because not everyone can hear the
Yeah,
question,
oh, sure.
can you repeat it?
Yeah, so the question was, given this multinational collaboration, how do will we be sharing this massive amount of data? And so the answer is we set up an FTP site where the data gets dumped, and we spend a lot of time just waiting for it to download. The good news is the first one I talked about, that's only in Florida. That's not Canberra, so that's not quite as bad.
Piggybacking on Sue's question there, then sharing with each other, FTP,
Yeah.
that's fine. How do you share beyond that, depending on your grant requirements?
Yeah.
and-
Yeah, well, that was part of our data management plan. This is an NSF-funded project, and so, yeah, and so all of the statistical analysis code that we're writing as part of this will be open source under the new public license. And right now we're hosting that on GitHub, but we'll have a link to use Figshare to get a DOI, so it's permanently referable to in terms of the code, and that'll be for relatively small amounts of data associated with the traits that we were talking about. So that'll work for that. The short read data that we're getting from all this genomics, NIH has a short read archive, and so all of the data will be submitted to NIH short read archive. And then we're of course setting up a project website that will have links to all of those things so that people can find them. If they find our project, then they can link to it, or they can, hopefully, NIH and the Figshare should have good metadata so that people can find it just through regular searches. Sure.
I'm just curious, these flowers are beautiful.
Yeah.
So assuming that people are cultivating them,
Yes.
and does that interfere at all with the wild? So that if they're cultivating for, big white flowers,
Right.
does the pollen get dispersed in the area, or is that an issue at all?
Mostly the places we're working, that's not an issue. There is one population, for one species we were sort of interested in. There's an area called Kleinmond, which is about 70 miles east of Cape Town, perhaps, where there's an area we were working, and there are cultivated protea in the area that we think could be potentially interbreeding with some of the ones that we were actually trying to study. And so we have to be a little careful with respect to the genetic analyses in terms of making sure there aren't protea farms nearby and those sorts of things.
But most of these areas are.
Most of the areas are relatively isolated, and it's not a problem.
And the pollen's not carried back.
No, so they're pollinated by birds,
Okay.
and the birds can move, reasonable distances. But the evidence we have from the microsatellites I referred to is that populations separated by more than a few kilometers, there's almost no movement. Yeah. There was another question here.
You answered it, the remote population.
Okay.
about how they're drastically different populations within a few miles.
Yeah.
Is that unique to like the terrain of that area? Because I would think that birds or winds or whatever would scatter.
No, well, no, actually, I mean, there's some controversy about this, to be perfectly honest, but my take on it is that there's, for most plant populations, there's relatively limited gene exchange beyond a few kilometers for sure. Now, the reason there's controversy is because there is some recent evidence that there may be occasional favorable mutations that are sort of globally favorable in a species, and those might, because they're favorable, sweep through a population much more effectively than the typical more limited gene exchange. But most pollinators, I mean, birds would move farther than bees or butterflies, but if you follow bees and butterflies around, they typically move very short distances, and so the distance that pollen moves is maybe on the order of only a few meters, much less, you know, kilometers or more.
Are hummingbirds the only ones that, for birds that pollinate are outer birds?
No, as a matter, there aren't hummingbirds in South Africa. I should have brought a photo. There are sunbirds and sugarbirds. They perform much the same role, but and then in Hawaii it's honey creepers.
Is there more diversity among the flowers that birds pollinate versus butterflies?
There aren't as many bird-pollinated flowers in general as insect pollen, and part of the reason is probably, now if you think about it from the pollinator's side, bees they need a lot of energy, but they're pretty small. So the flower doesn't have to produce a whole lot of nectar. But a hummingbird and sugarbirds are actually decent-sized birds. They have to produce a lot of nectar to make it worthwhile for the pollinator to visit. So there's a lot greater investment from the plant point to attract a bird pollinator than a bee pollinator. Yeah.
As you're doing research, do you ever feel not a pressure, but a need to communicate what you're doing in a way that the average person or your university community could appreciate, beyond maybe popularizing what it is that you're working on?
I try to do that to a limited extent. I don't think I'm terribly successful at it, but when I'm in South Africa, I do make blog posts occasionally. I'm tweeting I'm sort of tweeting where I am, and so there's a little bit of that. I don't think I'm terribly good at it, but I make a shot.
Is that something that you think institutionally people are asked to do, scientists are asked to do?
Institutionally, I don't think we've been asked to do it too much. I mean, certainly NSF is very interested as part of the broader impact criterion in making sure that those of us who are funded to do National Science Foundation work share that work as effectively and as broadly as we can.
And you teach it too.
Oh yeah. Less now that I'm a dean than I used to, but.
So in terms of the biodiversity hotspot
Yes.
that you showed earlier, what's the theory about why those areas as opposed to other areas of the globe? Because they have some obvious characteristics in common. They're mostly equatorial, but not entirely. They're mostly coastal, but not all coastal regions are.
Yeah.
So what's the current thinking there?
Well, so there's been a lot of argument. In general, tropics are more diverse than temperate or Arctic regions. Partly that's because, you know, as you move further north, we had, I mean, 10,000 years ago, we were under ice here, right? And so there just hasn't been time for new things to evolve here. So part of the excess diversity in the tropics may be related to more ancient lineages as having been able to persist there for a longer period of time. The Mediterranean climate regions are interesting. I suspect there, the ones, at least South Africa and southwestern Australia, have been geologically stable for a very long time. So they've been above water, unglaciated for, you know, billions of years. And so there's been a long time for things to accumulate there. They're also both sort of cut off from other places. So in South Africa, the Cape Floristic region is largely cut off from the rest of Africa, except for that narrow strip heading up east by the Kalahari Desert. And similarly, southwestern Australia is cut off from the rest of Australia by the Great Central Australian Desert.
so you don't have more successful species essentially migrating in and creating.
That and they're just isolated and sort of independent, and so evolve independent, and things will evolve and diversity will arise, and so they've just stayed there, and it hasn't gone extinct.
It also looks for the hotspot map, it's the places where the continents broke apart that this happens.
Well, it is coastal.
For the most part, not.
For the most part, yeah, but it's asymmetrical because Eastern North America broke apart from Europe, and we're not a hotspot. Yeah.
Just logistically, why are you choosing to collaborate with a university all the way in Canberra and not one in South Africa?
Oh, we actually are. I mean, we have colleagues at University of Cape Town and at the South African National Biodiversity Institute. The person we're collaborating with in Australia was originally at the University of Chicago when we were funded, and has since moved to Canberra.
Can you talk about the conservation controversy?
Oh, the hotspot? Yeah, sure. So, well, let me go. I'll just go back to that map because that's the easiest way to sort of illustrate it. Okay. So there's the map. And so the controversy arises because think about the way they produced this is they got species lists, and they didn't have, and keep in mind, we don't have a comprehensive species list for all organisms in the world. We have pretty good species lists for vascular plants, for birds, for mammals, reptiles, fish, and then butterflies, a few other groups. So they got the comprehensive species lists they could and identified the geographical regions that had the highest number of species in those groups and sort of that weren't represented elsewhere. So that's how they defined their hotspots. They're hotspots of biodiversity. And so in one sense, what they were arguing is those are the places where we need to concentrate our conservation efforts, because by focusing our efforts there, we can protect the largest number of species, potentially with the smallest amount of effort and money. Okay, so on the face of it, that's not an unreasonable thing to say. On the other hand, we also know, let's take something close to home, like the Connecticut River and the tidelands associated with the Connecticut River and salt marshes. We also know that salt marshes are among the most productive ecosystems in the world in terms, and they provide vital habitat for a huge number of fisheries and things like that in marine ecosystems, but they are not species diverse. And so if we focus all of our attention only on places that have high amounts of biodiversity, we're ignoring very important ecosystems that serve other functions. And so, as a matter of fact, to be a bit provocative, Peter Kariva, who's now the lead scientist for the Nature Conservancy, co-authored an article about 8 years ago now, maybe, in which he argued we should focus on cold spots rather than hot spots. Because some of those cold spots, like the Arctic tundra, like salt marshes, actually served more important biological functions than the hot spots did. And so that's some of another part of the controversy is, in fact, as I said, the hotspots were based on a list of a fairly small number of taxa. And if you look carefully, if you use those same lists of taxa and look just in the British Isles, where of course they've got everything cataloged down to very, very, very finely, you could actually put a grid over the British Isles and ask, if I define a hotspot using bird species, how likely is it that that's also a hotspot for lichens or for dragonflies? And the answer is it's almost not likely to be at all. That there's very poor correlation between hotspots in one taxon and another. And so this might represent the places that are most important for plants, birds, and mammals, but it's not the most important place, for example, for freshwater mussels. Any guess where that might be?
Great Lakes.
That would be right about here. The southeastern U.S. has a higher diversity of freshwater mussels than anywhere else in the world. I think. Sure.